Welcome, everyone. We're going to get started in just a minute or so. We're just letting all the participants into the webinar. Well, welcome, everyone. I'm Jen Farmer, the CEO at the Friedreich Ataxia Research Alliance, and I'm excited to kick off our second FARA Flash Talk series this evening in honor of FA Awareness Day and Month. Hopefully you all know that FA Awareness Day is May the 15th this year. We also not only celebrate the day, but we also use the month of May as really the opportunity to raise awareness. And, you know, when we think about awareness, we think about raising awareness, you know, in terms of the general public, helping more people know about FA. We also are focused on advocating for FA. And obviously this is something we do not just on this day and in this month, but throughout the year. But, you know, this month allows us to kind of come together as a community and circle around the importance of these awareness kinds of activities. It's also the opportunity to continue to raise funds for research. And importantly, and what we're here to do tonight is to educate ourselves further about FA. And I just wanted to share with you a few specific things before we get started on the Flash Talks. One is our campaign for this month is Lend Us Some Muscle. And the Lend Us Some Muscle campaign comes to us from our partners in Australia. FARA developed this campaign, gosh, I think about 3 or 4 years ago now, as a way to bring our global community together and raise awareness for FA. And it's a lot of fun. If you haven't participated yet, there's still an opportunity to. You can reach out to us. We'll send you some awesome tattoos. And all you have to do is tattoo, take a photo, and share it, just like Brendan's doing here. Our friends at the Burrow Hill Foundation have made a pledge to match $25,000 in fundraising, which is totally awesome. Thank you so much, Burrows Hill, and to all of you who are donating so that we reach our match. And the funds that we raise from Lend Us Some Muscle are going to a special research award that we've started last year, which is the Award for Innovative Mindset. And these grants go out to really, you know, kind of out of the box projects, really high risk, something highly innovative. And so it seems appropriate that, you know, our Awareness Month campaign goes to a special research project. But tonight we're here to focus on education. And so before we launch the Flash Talks, I wanted to make sure everybody also knew about the Minutes of Science. Minutes of Science are a new video series that FARA launched at the end of December. There are now 5 videos out. And these videos, in each video, we're sharing a little bit about a FARA-funded research project. And these are, you know, 3 to 4 minutes long. These videos are created by volunteers in our community, folks who are helping create the animations, the creative behind, you know, what the science that we're trying to communicate, as well as the narration is provided by our FA community members. And so these are nice, easy ways for you to learn a little bit more about the awesome science that you are supporting and helping make possible. And so with that, I'm going to turn it over to FARA's Director of Research, Liz Sarani, who's going to talk to us about the Flash Talk series and get us started this evening. Over to you, Liz.
Thank you, Jen. I'm Liz Sarani, and tonight we are going to hear 4 presentations from junior investigators. They're short presentation, only 5 minutes long. And they are examples of research that FARA is funding and of recent advances in FA research. We have challenged our young investigators to explain their work and to condense them in just one slide. But after each presentation, you will have the opportunity to ask questions during the Q&A session using the Q&A box that should be at the bottom of your screen. During the month of May, we'll have 4 sessions, including today's, every Thursday. Hopefully you have registered for all of them. But if you're missing some, the sessions are recorded and will be posted on YouTube. So let me tell you a little bit about what changes we've made this year. We have grouped the talks differently. Every session has several examples of different type of research that FARA funds. Some basic research on understanding the disease, for example, using cell and animal models, or examples of various therapeutic approaches or biomarker research. These are measures that can help us track the disease progression, and also examples of clinical research. Also, one new thing for this year is that we will recognize the best presentations with an award, and we will need your help. After the webinar is over, you'll be prompted to follow a link to a survey where you'll be asked to rate today's presentation on a few factors. I know this will be hard. I know that all the presentations today would be great, and every session will have great presentation, but we really appreciate your input on which presentation you like best. So please fill out the survey if you can. Also, I think we have put the link to the survey in a Q&A box. I don't see it at the moment, but I think we will put the link to the surveys into the Q&A box. And finally, I am very excited about this year because we have a co-moderator here with me to help with questions to our presenters. Our co-moderators are part of our FA community, and we're very grateful to them for volunteering to help. So before we start, it's my great pleasure to welcome Randy Yipe to the Flash Talks. Randy, would you briefly introduce yourself?
So we're the Yipes, and I'm Randy, one of them, a self-described FA science junkie. Very, very interested and incredibly excited to see some of the presentations tonight because they hit on some of the topics I think that we've seen at the FARA symposiums or the CHOP symposiums and some of the other scientific talks. So our children, Jake and Claire, both have Friedreich ataxia, and our other children, Katie, Ellie, and Tommy, are all in with us. So we Yipes are in and excited. So thank you.
Thank you. Okay, so now without further ado, our first presenter today is Rucha Dilip Sarwade. She's a postdoc with Dr. Balasubramanian at Monash University, and she's going to talk about investigating epigenetic silencing in Friedreich ataxia. Rucha?
Thank you, Liz. I'll just share my screen. Can we all see this?
Yep.
Okay, great. So good morning, good afternoon, good evening to everyone joining in from different time zones. I am Dr. Rucha Dilip Sarwade, and in the next 5 minutes, I will attempt to convince you all on why we must consider plants as the missing link that bridges the existing knowledge gap in our understanding of the pathology of Friedreich's ataxia disease. In case you're wondering, yes, this is my presentation for today. No, I promise I wasn't procrastinating. Yes, it will all make sense in the end, hopefully. On the slide, you will see 2 sentences that are trying to describe what's appearing on your screen. A slide. Let us try to reimagine these statements in terms of genetic code. So think of each of these statements as a stretch of DNA and each of this word as a gene. Now let's focus on the first statement. There are 4 genes in the first statement. The genes in the statement are doing a perfect job of explaining what's appearing on your screen. A slide. In the second statement, however, you will note that the fourth gene have a slight defect. This defect is hampering our ability of explaining what's appearing on your screen because of the number of times the letter I is repeated. Now, thinking about the human body, each cell in a human body contains 30,000 genes. A defect in one or more genes causes genetic disorders. Friedreich's ataxia is a product of a defect in a human gene called frataxin. This defect is caused by multiple repeats of 3 letters of DNA, that is GAA. This phenomenon of multiple repeats of 3 DNA letters is commonly known as triplet repeat expansions. The issue is that this is not a new discovery. We have known this for more than 100 years. A lot of research has been done since, but one of the most interesting discovery was made in 2009, where a team of researchers from Germany noticed that a plant that is widely studied in labs had irregularly impaired leaves and therefore was seriously ill at high temperatures. What's even interesting is that they found that this growth defect was also caused by GAA repeat expansions, due to which a plant gene was shut off, similar to what happens in Friedreich's ataxia. Historically, plants have led to several prominent discoveries. The discovery of cells, which is the basic unit of life, was first based on plant research. In fact, the discovery of genes and genetics was done in plants. So when you have found a certain disorder in plants that correlates with human defects, what do? Naturally, you want to dive deeper into the plant mechanism to try and understand their irregularities better, right? That's exactly what our lab did and has been in the forefront of this research. In 2018, our lab deduced the molecular mechanism of how repeat expansions led to defective plant growth. When the cell encounters a triplet repeat expansion, it switches the gene off. The cell then tags it with danger signposts all along the gene. This tagging mechanism is basically called epigenetic silencing. What's striking is that 8 out of 11 components of this silencing pathway that was found in plants has close relative in the humans. Perturbing any of these components in the human should effectively increase the frataxin levels and improve the diseased condition. That's where I come in. I am a molecular biologist with more than half a decade of experience working on cell lines. I became extremely fascinated learning about the similarities between plant and human triplet repeat condition. I'm currently testing the hypothesis that they indeed share a common underlying mechanism on cell lines that were derived from the patients and their unaffected siblings. In the next year or two, we intend to identify key players involved in the disease and come up with strategies to counteract them, thereby slowing down the disease progression. Once again, plant research has come to our rescue, and together we may all be on a path of a major breakthrough that may expedite our quest of finding a potential cure for Friedreich's ataxia. Thank you all for listening, and I'm happy to take any questions.
Thank you, Rucha.
So
Go
please
ahead, Randy.
feel free to post your questions. Rucha, if you'll allow me one.
Yeah, sure.
Do you have a thought as to the mechanism by which this triplet expansion repeat downregulates transcription of the gene in terms of your plant research or the analogs to human genes?
Yes. Yes, we do. So what has been shown in plants is that these triplet repeat expansions lead to something called as RNA-dependent DNA methylation, wherein these triplet repeat expansions are identified by the small RNA pathway, which leads to formation of small RNAs. Now, this small RNA then tags these triplet repeat expansion locus for epigenetic silencing. So therefore, the DNA methylation enzymes, the histone methylation enzymes are recruited at this locus and marks them with epigenetic marks and silence this locus. So we think a similar pathway is operative in the humans as well, and our preliminary results indeed suggest that some of that may be true. So yeah, we have some interesting unpublished data from the lab, which also suggests that SUMOylation, which is a kind of a protein modification, may be involved in this epigenetic pathway. And currently what I'm doing is trying to knock out, remove all these D-SUMOylases from the cells and see if it increases the frataxin levels.
All right, we've got some questions coming in. So one of the questions from an esteemed researcher in epigenetic silencing, who himself, I believe, is a fan of the methylation theory, asks the following: is it possible that plants are different from humans, mammals in general, in terms of epigenetic regulation of the repeats?
Yes, they are different. Like I said, that RNA-dependent DNA methylation pathway is actually only known in the plants. We don't know if it happens in the mammalian system or even in the humans. We have certain cues that we are chasing, and we have certain common links. Though the epigenetic marks are very similar in both the cases. So there can be the depressive and the excitatory epigenetic marks are similar in both the plant and the in the humans, but is this pathway or a similar pathway to RNA-dependent DNA methylation happens in humans is something we're not sure, we don't know yet. And yeah, we are mostly working on that.
So Rusha, maybe you can tell us why it's important to study also this mechanism in plant, and if there are things that we can do in plant that we cannot do in mammalian cells that can help us understand what happens in mammalian cells.
Oh yeah, that's a great question, Liz. So plant is, you can easily genetically modify plants, which is something not possible to do with humans or the cell lines that we have derived from the humans. And also the cell lines that are derived from the patients are actually not the ones that are affected. So I'm basically working on lymphoblast and the fibroblast. So they are not the ones that are actually affected in Friedreich's ataxia. So plants are a simple model organism that we are working on is Arabidopsis, that our lab is working on is Arabidopsis. And it is easy to genetically modify them and do something called a suppressor screening. So you basically do a large scale mutant screening and identify the genes that are responsible to suppress the phenotype. So the plants, these disease plants have a phenotype of, they look irregular, they look diseased. And if we do a mass scale mutation analysis on them and see if this diseased plant turn back or revert to something that looks natural or normal, those were picked up and seen where the mutations can lie. So this is something called a suppressor screen, and we can do such large scale studies in plants. So it is important to exploit the things that we can do in plants and use it as a starting material in order to understand the disease in the humans well.
Thank you. All right, I think we need to move on to the next speaker. The next speaker is Damon Wang. He is a grad student with Dr. John Watts at UMass Medical School, and he's going to talk to us about antisense oligonucleotides targeting frataxin intron 1 can activate frataxin expression. Damon.
Thanks for the introduction, Liz. Could you guys hear me and see the PowerPoint okay?
Yep.
Great. Hi everyone, I'm Damon, a graduate student in Watts Lab. I'm going to talk about antisense oligonucleotides targeting frataxin intron 1 can activate frataxin expression. So my talk will be divided into these 4 tiny squares in this single slide. First is I want to point out that oligonucleotide drug is a very promising drug modality. There are current over 10 oligonucleotide drugs approved by FDA. And taking Spinraza, one of the most commercially successful drug, as an example, with the chemical modification indicated here, either at the linkage or at the ribose sugar of their RNA monomers. This oligo drug is incredibly efficient and also stable after injecting into the central nervous system. They can be effective for the period of 3 to 4 months for one injection. And let's come back to the Friedreich ataxia. There's this very popular ORLU theory. As you can see on the top of this graph, because of there's a GAA repeat expansion during transcription, there's the stable DNA RNA hybrid structure formation, or ORLU formation, and that would further trigger a series of epigenetic changes such as histone deacetylation, methylation, as well as DNA methylation, which would further silence the gene expression that is the root cause of the disease. For the past period of 5 to 6 years, David Carr group have demonstrated that using oligonucleotides targeting the repeat region can restore frataxin expression in various cell models. However, there are huge challenges in his strategy in terms of low potency, as well as off-target concerns when we talk about drug development. So my hypothesis is, could I activate frataxin activation by targeting the non-repeat region inside of the frataxin intron 1 region? If you look at the top right panel, here comes with my design. So this is a zoomed in version of frataxin intron 1. The little red triangle indicates the GA repeat region, and each purple short line or the red line with the label as FA and a number, that is an antisense oligonucleotide I designed and synthesized in the same chemical modification pattern as the drug I showed you at the left top, very stable in the central nervous system. So with that design, I firstly tested those ASO candidates inside of the patient-derived fibroblast lines. As you can see here, I have several groups. The first black groups are controls, and then I have the ASOs targeting the repeat region in blue color, and targeting the upstream of the repeat region in purple color, and ASOs targeting the downstream in green color, and the last black colored bar is the wild type cells. As you can see, there's one candidate called FA9 here can significantly activate frataxin expression level at mRNA level, and that brings very close to the wild type level. Surprisingly, I also tested this FA9 candidates in wild type cells. It can also further increase the frataxin mRNA level at wild type cells, indicating this action is independent of the repeat expansion. From there, I also tested a series of cell types, including cells from liver, kidney, as well as glia in the brain. All of them, I can get significant 2- or 3-fold activation. I mean, in the process of testing in neuronal cell models, and this indicates that this activation mechanism is widely conserved across from different cell lineages. My future direction includes, at the bottom, I need to validate the frataxin activation at the protein level. Also, I need to validate these ASO candidates in the disease relevant cells, such as neuronal cells, as well as cardiac cell models. Meanwhile, I need to understand the exact mechanism of the action of these ASO candidates, whether they work at transcriptional level or post-transcriptional level. And I also want to show my appreciation for the tremendous support of FARA community, and I will look forward to further exploring the potential of these drug candidates and push it closer to potential clinical study. Thank you very much. I'm willing to take any questions you might have.
Thank you, Damon.
That's fantastic research. Everyone who's familiar with Spinraza knows that that's something of a miracle drug, and it's really changed people's lives. Do you see a translation of some of this research in antisense oligonucleotides to FA? And I think a corollary question would be: do you see the length or the varying length of the GAA repeat in different individuals with Friedreich ataxia as a challenge to the deployment of antisense oligonucleotide therapy?
Right. Thanks for the question. As far as the length issue, we tested our oligo candidates in two fibroblast lines. The first is shown here with about 300 G repeats, but also tested a second line with over 500 repeats. I also got significant activation there. In terms of the translation, I see there's definitely the potential there, but we do need to do more work before reaching that stage. And those work include that we need to make sure that our ASO candidates work in the patient-derived neuron cells from the reprogrammed stem cells. And that would be the key evidence telling us whether we can go there or not.
One of the questions from our audience, do you have a sense, or do you know if this oligonucleotide disrupts the R loop?
Yeah, that's a great question. So there's a paper in 2014 kind of mapping the R loop area in frataxin locus, which was not limited to the G repeat expansion region. And so this FA9 candidate indeed is within the high R loop formation region, while we have also a second candidate which is outside of the higher R loop formation region, literally in the middle of the intron. We do not know yet, but we are definitely interested in using some of the antibody-based strategy to evaluate whether this really reduces local R loop formation.
And can you comment your research on FA9, that particular oligo? The comment that you made that you tested it in the wild type cells and it actually upregulated, it increased frataxin production in regular cells. Can you comment on the location of this oligo being upstream of the GAA repeat and how that would relate to the upregulation of frataxin production in wild type cells?
Right. So as I said, this FA9 is within the higher R loop formation region. I think it's probably around 300 BP upstream of the GAA repeat region. As far as you mentioned now, why it also further increased frataxin mRNA in wild type cells, it is surprising to us. We haven't fully understood it yet, but one possibility is that this RNA region that FA9 is targeting serves as a regulatory role in gene expression. It is also present in wild type cells.
We have another.
Go ahead.
No, we have another question from the audience. Ron Bartek, as some of you may know, founder and president of FARA, co-founder and president of FARA, asks, which cell lines do you plan to use in testing neuronal and cardiac cells?
Right. So for the neural cells, I'm in the process of testing a wild type background, the SY5Y cells, because that is relatively easier to culture. I think the golden standard here for should be the iPSC derived neuron cells and cardiac cells with the GA repeat expansion. We don't have much good access to those kind of cell models. And And I would like to point out here, if anyone is interested in working on this, potentially, we are very interested in potential collaboration.
Great. Just one quick question. Do these oligos bind the mRNA? And do you know if their effect has some effect on the mRNA stability or degradation?
Yeah, that's a good question. I think that's in my future directions for understanding its mechanism. So I think it would as designed binding the intronic RNA in the nucleus, but we don't know yet whether it worked at the transcriptional level or not. So with one cheap experiment to figure it out is using small molecule to inhibit global transcription and treat with our ASO, see whether it still works or not. If it still works, that might indicate it potentially works by stabilizing the mRNA.
This area of the gene where you tested FA9 and saw this remarkable response, do you have any sense as to where that would, where in the gene that would be relative to, these higher areas of methylation that we see with the GAA repeat in the CPG island shore or any of that?
I'm not sure about the CPG island shore, but one interesting point is that I think Dr. Sanjay recently published a paper about the DNA methylation areas in the introns of the frataxin. And interestingly, this FA9 falls into one area with hyper DNA methylation formation. So it's potential that, this FA9 might kind of interfere the local DNA methylation patterns and thus activate the frataxin expression. But those remains to be tested.
Excellent.
Okay, thank you, Damon. I think
Thank
we
you.
need to move on to the next speaker. So we're going to switch gear completely. As I said at the beginning, we're going to have a different example of research in FA, and now we're going to switch to more clinical research. Our next presenter is Jacqueline Tamarov. She's a fellow in the Division of Endocrinology and Diabetes at CHOP with Dr. Shannon McCormack. And she's going to talk today about NAD and exercise in Friedreich's ataxia.
Switching gears is a great pun, by the way.
Not intended.
There I go. So hi, I'm Jacqueline Tamarov, and I'm a third-year pediatric endocrinology fellow at the Children's Hospital of Philadelphia. I'll be speaking to you about an actively recruiting NIH-funded clinical trial called NAD and Exercise in FA. I started research in FA through my mentor, Dr. Shannon McCormack, and have become interested in better understanding endocrine disease in FA. I've also really enjoyed working with patients and participants with endocrine disorders related to FA, including diabetes, growth concerns, and bone health concerns. This trial was really designed thanks to questions that you asked during the FA symposium. My mentors, Dr. McCormack in endocrinology, Dr. Lin in cardiology, and Dr. Lynch in neurology, all have been asked about exercise in FA. We know it should be beneficial, but how much? What type? This has never been systematically studied. At the same time as these questions were arising, there's been recent interest in NAD, which is important for energy production in the cell and may enhance the exercise effects. This has also not been evaluated in human studies in Friedreich's ataxia. With this, our team and the help and support of FARA proposed to the NIH that we study these interventions, exercise and NAD in Friedreich's ataxia. The goal of this study is to see how exercise and NR, an NAD precursor that can be taken as a supplement, can benefit fitness, which is the body's ability to use oxygen during exercise, and glucose or sugar metabolism. Our hypothesis is shown in the figure on the left, and it is, do these interventions improve mitochondrial function and increase fitness and increase insulin sensitivity, which is a way to measure your body's response to glucose? This is a randomized placebo-controlled trial that's going to recruit 72 individuals over 5 years in total, ages 10 to 40. So far, we've enrolled 10 individuals, And I'd really like to pause for a moment and thank everyone who has enrolled, participated, or shown any interest in our trial. We really appreciate it. So individuals will be assigned to 4 groups randomly, either exercise or no exercise, and either supplement or placebo, which is a capsule with no active ingredients. The study involves 2 in-person visits over 2 to 3 days at the Children's Hospital of Philadelphia, and they are 12 weeks apart. During this study, we're evaluating your fitness and your glucose metabolism using multiple mechanisms. These include an exercise test to look at fitness, a DEXA scan, which is an X-ray that looks at bone mineral density or bone health and muscle mass, a leg MRI that looks at mitochondrial function, and an oral glucose tolerance test that evaluates glucose and insulin sensitivity. We also are going to place a continuous glucose monitor, which is a really exciting research tool since it can evaluate glucose over 14 days and is worn home from that study visit. Between the visits, so while you're at home, the exercise group will do exercise 30 minutes a day for 5 days a week. 3 of those days will be trike, and 2 of those days will be strength training. And so here is Kyle Bryant, whom you all know, on his trike. And I'd just like to pause for a moment again to thank Kyle, because if he had not been involved in this study and without his insight, we really wouldn't have been able to do this study. So the trike, unlike Kyle on the street, will be on a trainer at home, and we provide it for the study. We also have an app that tracks your exercising at home, and that was designed by the cardiology division with this study as an impetus. All groups will take a capsule once daily, either the supplement or the placebo. And we know capsule size is important to some folks. And so I showed you the capsule here next to some candies, Skittles and Mike and Ike's, to give you a sense of the size. Similar, maybe a little smaller than the Mike and Ike, but a little bigger than the Skittle. So what are some common questions that we've gotten about this study? Well, it's 12 weeks with 2 in-person visits. Everything else is done at home, and travel is reimbursed to CHOP. It is okay and even great if you don't exercise now, but at this time you must be able to sit unassisted and pedal on the recumbent trike. We are in the future hoping to add an option for those who cannot pedal. Since we know that people really want to do this study because of the exercise, if you're assigned to the no exercise group, we provide the same intervention to occur after the study if you would like. Also, you can continue prior exercise or physical therapy in any of the groups, whatever you were already doing. And lastly, you're given the recumbent trike to keep. If you're interested in this study, please reach out to our research coordinator, Anna Dedio. Her email and phone number are here on the slide, dedioa@chop.edu, or talk to any of your doctors or research coordinators, and they can certainly put us in touch with you. I'd really like to thank my mentors, the NIH, for funding this study, and of course, FARA. And I'm really grateful to be involved in this community and partner with patients in research. I'm currently supported by a FARA postdoctoral fellowship award, so thank you. And I recently was awarded an NIH grant to better understand the intersection of blood sugar and heart disease and Friedreich's ataxia that I'm really excited about. So thank you all, and I'm happy to take questions.
This is great. Jacqueline, looking at your slides, do you think that exercise has an effect on just the overall number of mitochondria or on the efficiency of those mitochondria in the subjects that you're looking at?
I believe that our thought is that it will be mostly on efficiency of the mitochondria and oxidative phosphorylation, which is sort of the mitochondrial's function itself.
Some questions from the audience. How do you, in this study, given that some of the exercise is done at home, how do you monitor compliance among the participants? That is everyone actually doing 5 days a week on the bike and with the strength training?
So great question. Compliance with the study supplements is an easier thing to monitor. So we use a Clever Cap, so that helps us see if you are taking the supplements. And then we also look at the amount that are left over at the end. So that's sort of commonplace. For the exercise, we have an exercise app again. And so that uploads to our computer system, and we're able to see, that you had that increased heart rate during the exercise. The app is attached to a Fitbit.
And what is the dose of NR that you're using in the active arm of the study?
You know what? I am actually not sure about that, but whoever asked that question, I'm happy to find out and to send you an email or follow up.
And if I took FR, does that mean I don't have to exercise any longer? That's
If you
not.
NR, do you have to exercise?
No.
So we do think that both of them are probably very helpful, but probably even more helpful together, or that's what our hypothesis is, and that's what we're testing.
One of our audience members asked or stated, your picture reminded me that my son with FA loves Skittles and Mike and Ike's. Is sugary candy common in FA patients? And if so, is there a scientific reason why there is a sugar craving with FA?
So I have not heard of the sugar craving specifically. I'm sure others might have more insight. It's not something that I've been told. And yes, I know I as an endocrinologist, feel like I don't know that I should have put candy pictures on there, since most of what I deal with has to do with sugar-related concerns. But it is really helpful just to get a sense of the capsule size.
Others are curious if you expect to see a benefit of exercise on the nervous system, on the function of the nervous system as well.
So our main hypotheses are really that it's going to affect how well you exercise, which makes sense and sort of is what intuitively we would think, and how well you and your glucose metabolism. There are a few extra things that we did not include here, like a FARA score. And so we are certainly looking at the nervous system. How much, that over 12 weeks may be difficult to see anything. And so that's not something that we're primarily studying.
After the study ends for an individual, is there any support system or any plans to kind of continue to encourage participants to exercise after the study concludes?
So that is a great question. So for the no exercise group, we offer like the full 12 weeks with us calling you to follow up, us increasing your exercise prescription. After that, I believe there's been discussion over whether there may be a clinical ability to still use the app, at least for those followed at CHOP, though that's not so up and running yet because the app was actually developed to be used both in clinical use and research, but the impetus for getting it going was this study, so it's what it's being used for first. And you get to keep the trike, so we hope people are continuing with exercise, but it's definitely with us a little bit less involved.
And that bears repetition. Those who participate in the study get to keep the trike as their own afterwards. Yeah,
I think that's really one of the most amazing things about this study. It's awesome. And the trike we give you, we only have you use for the study on the trainer, so that the people who live in a really hilly area and the people who live in a really flat area don't have different experiences, or the people who do it in the winter and the summer. So we do it in the home, but it does not have to be in the home. So you can use it outside after the study.
There's another question here. I think the point of the question is one of the outcomes of your study meant to confirm like mitochondrial biogenesis, or is it meant to demonstrate some sort of a synergistic effect of the increased number of mitochondria in the treated group, the folks with the NR supplement?
So it is our thought is that we may find a synergistic effect. We may find that both exercise and NR together is what best improves the fitness, which is sort of what we're using to demonstrate that the mitochondria are working better.
And within a reasonable degree of scientific probability, can you offer me the following: that if I exercise, I will become as handsome and clever as Kyle Bryant? Or is that just that's not attainable? That's a horrible question.
That one's a tough one for me.
No, we're not. No, I know the answer already.
On that one, I think we're moving on, right?
Thank you so much for all of the questions, and to whoever asked about the amount of supplement, I'm happy to follow up with you. I'll follow up with FARA to let you know.
Yeah, I'll send you the name and the email, Jacqueline. Thank you so much. Okay, the next speaker is Louisa Salvadore. She's a research assistant with Dr. Giorgio Caristianis, sorry, at Monash University, and she is going to talk about brain white matter pathways in FA.
Thank you so much. Hi, everyone. So my name is Louisa Salvadore, and I'm a researcher at Monash University in Melbourne, Australia. So I started working on FA research as an undergrad honours year student, and I continued my research as part of my doctorate in clinical neuropsychology, which I finished last year. So today I'm really excited to share with you some highlights from my doctoral research, which was focused on brain white matter pathways in FA. So my research focuses on human brain imaging or brain scanning in FA. So brain imaging is important in FA research for a couple of reasons. First, it helps us to understand which specific parts of the brain are impacted in FA so that we can better understand the disease process. And secondly, we can obtain a number of objective brain measurements, which are potential useful biomarkers for tracking disease progression and in clinical trials. So we know that good biomarkers are essential in the process of working towards a cure. So these are some of the aims behind the IMAGE-FRDA study from which my research comes. So this was a single site study conducted in Melbourne, and it involved 36 adults with FA and 37 matched controls who came in and participated in a baseline study and then came back for a 2-year follow-up. So the participants went into the scanner at Monash Biomedical Imaging and did a sequence of different brain scans. Now, this gave us different types of brain scans, which we then analysed to obtain different types of measurements of different brain properties. Now, my particular research focused on the white matter of the brain. So these are the connections between different parts of the brain. So think of the white matter as the wires that are transmitting and sending information between different parts of your brain. So different types of brain scans gave me different information about things like how much white matter there was and also how well the white matter is organized. I was then able to compare these measurements between people with FA and the control and also track how these measurements were changing across the 2 years of the study. So I found that several areas of white matter were affected in FA and that this seemed to reflect changes in both how much white matter there was and also the organization of that white matter. Now, when I looked at changes in these brain measurements over time, I found a particularly interesting finding that I've illustrated here on the slide. So what we're looking at here is the relationship between the 2-year changes in the amount of volume of these white matter areas and the participants' baseline Friedreich Ataxia rating scale or FARA score, which measures disease severity. So what the top row here is showing is that amongst the individuals in our study that had a higher disease severity at the start of the study, they seem to be showing more decreases in white matter in the cerebrum or the main sort of upper area of the brain. However, as shown in the bottom row here, amongst those individuals that started the study with a lower disease severity, we were seeing more of these volume changes in more of the cerebellar and brainstem regions. Now, this raised the concept of white matter changes happening in different parts of the brain at different stages of the FA course. So in this case, it's suggesting the cerebellum may be affected earlier on, and then the cerebrum or the other main parts of the brain later on. Now, this finding has important implications when we're thinking about biomarkers because it's showing us that one size may not fit all. So in other words, the part of the brain that's the most useful biomarker in terms of tracking, changes and progressions in the disease may be different depending on the person and what stage of FA they are actually in. So this single site work has been really important for identifying that brain imaging can provide us with really useful measurements in FA and showing us potential brain regions that we can take forward to focus on in future work, including in the important task of developing good new biomarkers. This work is now being extended in a multi-site longitudinal international study called TRACK-FA, where we can take advantage of a much larger sample size to dig much deeper into some of the patterns that we've seen in this type of work. And in terms of what's next for me, I'm actually soon heading over to Boston to join a new research team, and I'll be focusing more on my clinical neuropsychology training and looking at how ataxias can affect people's thinking and their mood. So I'm really excited to continue researching in this area, and thank you so much for the opportunity to present today.
Louisa, I see that you've correlated FARA score and disease severity with the amount, the volume of white matter. I know that we know from prior research that FARA score is related to GA, or I guess age of onset is related to the length of the GAA repeat. Have you ever tried to tie the two together, looking at age of onset and the varying length of the GAA repeat versus the amount of the volume of white matter?
Yeah, so as part of my study, my correlates that I used were the FARA score as well as the GAA and the age of onset. And here I'm looking at how those correlate with the amount of change across the 2 years. So my finding about the FARS was the key finding, but there were also some patterns, sort of less consistent in terms of sort of the pattern and the story that's there. But those are also correlating with the amount of change. So what it's really telling us is that there's heterogeneity, or that there's different trajectories for different people. And these factors like the disease severity, the onset age, and the repeat length are all factoring into people's different experiences in the brain. And I guess what this is showing is that not one size fits all, and bigger studies are so important for showing how different people may have different trajectories, which is really important for understanding what's the best biomarker for an individual, and also what is at the more nuanced level, what is the disease process actually looking like for different people.
You mentioned the white matter was akin to like the wiring of the transmission. Could you give us like a 101 course on the difference between white matter and gray matter?
Yeah, certainly. So our brain is made up of many different types of brain cells. And the key one, one of the main types of brain cells is the neuron. And the neuron is a cell that transmits information in our brain and processes information. It's got a cell body, and it's got a tail, or what's called an axon. So the cell body has the nucleus, and there's a processing happening there. And the axon is like a wire that connects it to another neuron so that information can be passed on. Now, the wire or the axon is actually coated in a fatty substance called myelin. And that's like an insulator to make sure that the message goes through effectively. And that myelin, that substance, has a white color. So when you actually have a look at a brain, that area or those parts of the brain where all these wires are appear white because of this fatty sort of myelin sheath that's coating the wires. When we look at other parts of the brain where more of the cell bodies are concentrated together, and these are sort of the areas of the brain where the processing of the information is happening as opposed to the transmission or the sending of information, those appear by contrast gray when we look at a brain slice or something like that. So what we would see in the brain is around the outside of our brain is what we call the cortex, which is where all the gray matter sits. And then underneath that, you can sort of see in some of my pictures, it's whiter, and these middle interior parts of the brain are where all these wires are crossing together.
Do you have any sense as to whether the decrease in volume in the white matter you're seeing is attributable to like to atrophy or some sort of a demyelinization process associated with the disease itself?
Yes, that's a really interesting question about what's the mechanism. Now, what we've looked at here is we've taken a brain image, and we're essentially looking at values of different pixels in the image to infer things about what's happening, but we're not looking down at the level of what's happening at individual axons and seeing what's happened. What we do know is that different types of brain scans give us different information, and they're about whether it's sort of more of a myelin effect or if it's a volume effect. And when we compare these different scans, it does look like there could be some areas that are affected, particularly with the myelin. But we can't really be very confident with that until we look at more of the histology and those kinds of things.
Yeah. A couple more questions. The period of time in your study, 2 years, is a long interval to wait in a clinical trial, especially with a progressive disease. Are there any changes in FA white matter that can be seen in a shorter period of time?
Yes, so it's not something that we were able to look at with this work, but I'm definitely aware of some work that's been done in Minnesota, and they actually found changes in more of the spinal cord areas, which we know are really impacted in FA as well, that were showing some changes across, more of a 1 year time span. And the really exciting thing about the next larger multi-site study, Track FA, which is kind of building on this work and a lot of other work around the world, is that that's actually going to have a baseline, a 1 year follow-up and a 2 year follow-up. So we're going to be able to improve on this and get what's actually changing at that shorter time scale.
There have been a number of presentations and some research done about the impact of cerebellar insult on movement and coordination in FA. Can you characterize at all the impact of this reduction of white matter you see in the cerebrum and what symptoms you'd expect to observe in such patients?
Yes, it's a really excellent question in terms of what does this actually mean for symptoms. One thing I will point out is that we're seeing these white matter changes or effects by comparing, a reasonably large group of people with FA compared to those without. So we're seeing that at that level. When I would pull up an individual person's brain, say someone with FA, it's not going to be an obvious change that you might see in a clinical scan. So it's not at that level. But we are seeing these maybe more subtle changes that can be identified when we compare groups together in a quantitative sense. It's really hard to pinpoint exactly what effect that may have, but we do know that, our whole brain works as a network. In any type of task we do, all of these brain connections are working and transmitting information. Some things that can be affected when white matter is impacted could be the efficiency of our thinking tasks or how quickly we can do things and things like that. But in FA, there's no particular, lesion or sort of severe reductions that we can say at an individual level. This is how this is going to affect someone's life. So something that hopefully as we, in studies like Track FA, we're actually collecting a lot of cognitive data as well, where people do some tests, and hopefully we'll be able to correlate those with the white matter changes to actually find out what is their clinical significance.
Great.
Yeah.
Thank you. Thank you, Louisa.
Thank you so much.
So let me just, since you mentioned something that oops, let me share my screen. There it is. Okay. Since you mentioned Track FA, I just wanted to spend the last minute talking about this project. You've heard from Louisa how researchers are using this sophisticated imaging system to take pictures of the brain and the spinal cord and to analyze them. And we'll hear more about this in the next sessions. But FARA has just funded a new study that Louisa mentioned that's called Track FA, that tracks the changes in the brain and the spinal cord in FA and can help researchers monitor disease progression. So it is done by international consortium of investigators, and they're recruiting 200 adults and children with FA and 100 controls. It'll consist of 3 visits, one baseline, one after 1 year, and another one after 2 years. And it will consist of a scan and a few tests, and also blood collection. So you can go to the FARA website to learn more about it, and look for communications from FARA on this probably in the next month or so. And this is a very important study, so please consider participating. Finally, let me thank the speakers again for all the great presentations today. Randy has been a fantastic moderator. And remember to fill out the survey and vote for your favorite talk, flash talk, if you can, after the webinar is over. And we hope to see you next Thursday at 12 Eastern. Thank you all for participating.
Outstanding thoughts. Thank you, guys.
Thank you.